So, what's sweeter than a woman's kiss?
A man's kiss.
What's sweeter than a woman's kiss? Glucose, because I need it for glycolysis, right? We watched the videos? No, we didn't watch the videos. If you're at a loss to what I'm referring to, the video on glycolysis takes the Archie's tune Sugar Sugar and changes the words but sings about glycolysis to that tune. It's really entertaining. I recommend you take the time to see it or listen to it anyway. Okay, so we were talking about metabolism last time. Just as a matter of review, we'll go through it one more time. Remember, it's a series of pathways, and we could start by talking about glycolysis, which we take glucose, a 6-carbon molecule, and we break it down into 2 3-carbon molecules called pyruvate. So glycolysis takes some energy. If you remember, we have to put in a couple of ATP initially to get this process started. But when we are done, we're going to get some reducing power and we're going to get some ATP, aren't we? Almost all organisms are able to carry out glycolysis. Even those that are fermenters, that don't use oxygen, don't have a terminal electron acceptor that will allow for respiration, carry out glycolysis. Now, this pyruvate molecule then undergoes a transition, if you will. It's converted into a 2-carbon molecule we call acetyl-CoA. Actually, the 2-carbon molecule is acetyl, but a cofactor hangs on just kind of as a carrier, if you will, to transfer this acetyl group. But this is a 2-carbon molecule, and so we've given off, a carbon molecule, and we've generated some reducing power, haven't we, in this? And this happens twice. We have to remember this transition step happens twice. Then the acetyl-CoA enters into our tricarboxylic acid cycle, a cyclic pathway in which we continue to break down of the glucose molecule. So this 2-carbon molecule here joins up with a 4-carbon molecule to produce a 6-carbon molecule in the cyclic pathway. And then, if you remember, we throw off a carbon molecule, and in doing so, generate some NADH, some reducing power. We throw off another one, generate some NADH, some reducing power, and at this point, we have completed the breakdown of our first pyruvate molecule. We can continue with some more production, some ATP, some FADH, another molecule of reducing power, and then the second pyruvate molecule enters into the cycle. And we'll turn the cycle one more time. And so for every glucose molecule, we're going to turn our TCA cycle twice to complete the breakdown of that glucose molecule. And so now we've pretty much completed the metabolism of that glucose molecule. We've captured all the energy that there is, really. Now we've got to convert that energy into ATP. And that's where cellular respiration comes in play. Remember, on our membrane, on our cell membrane, in the case of prokaryotes, we're talking about the outer cell membrane. They don't have any inner cell membranes. In the case of eukaryotes, we're talking about the inner cell membrane of the mitochondria, where we're going to see these proteins embedded into the membrane here, and they serve basically to pump hydrogen ions out of the cell to create a gradient. And we have carrier molecules that will transfer those electrons from one protein to another. So we have now these molecules that are going to be high in reducing power, the NADH molecules that have been generated from these two previous pathways. They're going to come over here and they're going to drop off those electrons that they have, and in doing so regenerate our electron carrier. And that's very important that our electron carrier is being regenerated. They have to be regenerated in order for these other pathways to continue. So now the electron's been passed off, if you will, and there's two of them that get passed off, and we see then that the energy from that electron is going to be used to pump hydrogen ions out of the cell. And so this electron will be picked up and carried over to this complex, picked up and carried over to this complex, and so forth. And each time it gets passed from one complex to the next, we are going to pump hydrogen ions out of the cell, continuing to create our proton gradient. Our proton motive force is being developed here. Again, pumping hydrogen ions out of the cell. But, as you remember, at the end of this, if you think about it as a stair-step pattern here, at the end of it, if our electron is high in energy up here, with each pass, it loses some energy. And so at the end of it, it's spent. There's no more energy there to capture. And so our oxygen molecule picks it up, becoming water, and allowing the pathway to continue. So this is cellular respiration. What is being generated is the proton motive force. And the process of ATP generation that's going to take place, when the hydrogen ions move back into the cell, we're going to capture that energy in the form of that high energy phosphate bond that we can now use as a currency. So the hydrogen, the proton motive force generates this gradient that will allow these hydrogen ions to move into the cell to generate our ATP. This is oxidative phosphorylation. ATP generated over here was by substrate phosphorylation. If oxygen is not available, this electron carrier is not regenerated or recycled, if you will, and so it is not able to pick up the electrons in these pathways, and so everything shuts down if oxygen is not available for this cell to pass that electron on. So oxygen in this case serves as the terminal electron acceptor. Now in some cases, some microbes have the ability to carry out respiration anaerobically. So they're using something other than oxygen as their terminal electron acceptor. Sulfate or nitrate, something like that, is serving as the terminal electron acceptor. Any questions? Yes?
Does that say half of an oxygen?
It does. Because, see, an oxygen molecule doesn't exist by itself. They always come in pairs. So you see I have two oxygen molecules, and so only half of that means one oxygen. Okay, but they don't exist individually, really.
So then it only releases one H2O. So one H2O for each time.
Yes. Other questions? Okay, so let's say we're out of oxygen and we're an aerobic organism and we need to continue to produce energy. Well, that's where fermentation comes into play. Fermentation really is taking pyruvate accepting the electron from NADH such that it can regenerate the electron carrier, and in doing so, gets converted into lactic acid. Well, not always lactic acid. There are a number of end products that some bacteria can make. We talked about alcohol, or ethanol, I should say. There's a middle step where acetaldehyde is formed first, and then the electron is received before ethanol is formed. Other things, acetone is formed, acetic acid, propionic acid, foamic acid. These are all different end products of the fermentation process. But it's all the same, and the purpose is all the same, in that we are just trying to recycle these electron carriers. Why? So that glycolysis can continue. And that's the only thing that's going to continue with fermentation. This has been shut down because there's no electron carrier there. Oxygen's not available to receive that terminal electron. So this is shut down. And this would shut down too, except for using pyruvate to regenerate this electron carrier, we can continue glycolysis. And so we don't get a lot of energy. You know, you only get 2 ATP really from the process of glycolysis here. But that's enough to allow the cell to continue. And so like E. coli, for instance, will use fermentation when it doesn't have a terminal electron acceptor. In this situation, we can say that glucose is partially metabolized because we're just breaking it down into 2 6-carbon molecules that then will become, from 1 6-carbon molecule into 2 3-carbon molecules. Now in your own cells, when you do this, you produce lactic acid. And that lactic acid then makes its way eventually back into the bloodstream to the liver, where it gets converted back into glycogen. And so you really don't burn a lot of calories when you have an anaerobic condition develop, because you've gone into just breaking down the glucose into pyruvate through glycolysis. The thing that we want to emphasize here, pyruvate is serving as the terminal electron acceptor in this case. Any questions? Okay, so that pretty much covers what I'm going to talk about in chapter 6. The chapter goes on to talk about some of the other metabolic pathways that are used. Photosynthesis is discussed, and we obviously know that cyanobacteria and algae would be using the photosynthetic pathway. Photosynthesis also establishes a proton motive force, or a hydrogen gradient, but instead of using chemical energy to do so, so instead of breaking down glucose to do so, they use sunlight to help them establish that proton gradient. Any questions about chapter 6? Yes?
So you said prokaryotes and eukaryotes do the different thing in the cellular respiration, right?
Yes, okay, so let's imagine here, this is my prokaryotic cell, okay? The proteins that are embedded in the membrane are going to be right here on this prokaryotic cell, embedded on those membranes. If we were to look at a eukaryotic cell, so this is a eukaryotic cell. Inside the eukaryotic cell, we'll find the mitochondria, and then the proteins for respiration are found on that membrane of the mitochondria. Questions? Okay, so we're going to move on to bacterial genetics, our last topic before our exam, which I guess is scheduled for next Monday, correct? We have, am I surprising you with that announcement? No, it's, I think it's next Monday. We are doing review and catch up next Wednesday, or this coming Wednesday. Well, before I get into Chapter 8, Bacterial Genetics, I want to do a little bit of review from Chapter 7. Now, I'm not going to go too much into Chapter 7. I would expect that you've already been exposed to some introductory genetics, I should say. So I'll give you a little bit of that. If you need to, you might want to go back and review Chapter 7 for your own sake, if you're not completely comfortable with genetics. So we'll start out by saying that our DNA, our genome, is composed of 4 nucleotides, adenosine, cytosine,